Welcome to the Environmental Mass Spectrometry video series. We are the Laboratory of Environmental Chemistry, Graduate School of Science and Engineering, Kagoshima University. In this video series, we will explain our manner to determine the molecular formula of unknown chemical contaminants in environments. Each video is about 10 minutes long or so. So let's get started. Mass spec is the clipped word of mass spectrometry. Also, you can say it just MS. But please be careful not to say mass. MS is the abbreviation of mass spectrometry, so you have to say MS as its abbreviation. First, why mass spec? Actually, we have several reasons, but mainly three. Sensitivity to detect trace contaminants in the environment, selectivity to distinguish resemble contaminants, and structural elucidating power of unknown contaminants. These three features are indispensable for environmental chemistry. Also, these features may be useful in your own research besides environmental chemistry, like biology, biochemistry, pharmacy, organic or inorganic chemistry, food safety and so on, so please enjoy this video series. Before we get started to learn mass spec, let us introduce environmental chemistry. Do you know how many chemical compounds are utilized in your country? In Japan, according to the Chemical Substances Control Law, 12,958 chemicals are utilizing over 1 ton per year, in manufacturing smartphones, computers, detergents, medicines and so on. Our comfortable and safety daily life is strongly relied on a variety of chemicals. The problem is, some of them or many of them are toxic against human or wildlife. Important thing is that we must take a balance between the benefits and risks of chemical usage. To take a balance, we must control their exposure to human and wildlife. Once you fail to control them, some adverse effects will appear sooner or later. But how? The answer is risk control. This is a typical workflow of risk control. First, you must identify which kinds of adverse effects are posed. Second, quantify the relationship between dose and effect. Third, assess exposure amounts and pathways, outcomes and populations. And fourth, estimate the overall risk to human or wildlife considering uncertainty. These 4 steps are so-called risk assessment. And then, determine how best to protect human and wildlife with the most appropriate way. In these steps, exposure amounts can be determined by environmental monitoring. That is the reason why so many scientific papers are published across the world every year regarding environmental contaminations in river waters, lake water, sea water, potable water, sewage, industrial effluent, and so on. Environmental monitoring is the processes to characterize and monitor the quality of the water environment, the air environment or the soil environment. Particularly freshwater environments have been extensively studied for many years, and there is a robust understanding of the interactions between chemistry and the environment across much of the world. Also, environmental monitoring can be classified in terms of substance to be monitored, namely chemical monitoring, biological monitoring, etc. In this video, we are focusing on chemical monitoring in freshwater body. In most cases, chemical contaminant concentrations in water samples are trace compared to coexisting natural organics. So concentration and purification processes followed by sensitive and selective analyses like mass spec are needed. This is a summary of sensitivity data of major detectors, cited from a Japanese industrial standard. Among these variety of detectors, mass spec is highly sensitive compared to the others. Mass spec can detect 10 femtograms. However, UV-Vis near detector required 10,000 femtograms for example. Please be informed that femtogram is to the -15th power gram. You may able to measure 10 mg by using a balance in your lab. But you may not able to measure 1 mg. By using a cutting-edge mass spec, 0.05 femtogram of a herbicide mefenacid can be detected and quantified quite satisfactory. Surprisingly, the number of mefenacid molecules were about 10,000. Sensitivity of mass spec is pretty good to determine environmental contaminants. This is the first reason why we are focusing on mass spec. The next reason is selectivity. Selectivity can be defined as the capability to distinguish or choose proper analyte among resembles. If you quantify impurity in your experiments instead of the analyte you focused on, that is terrible thing. Mass spec can select proper analytes with different masses. For instance, mass of the chemical on the left side is 202, and that on the right is 157. Mass spec is inherently selective for different masses. Even though analyte and impurity are the same mass spec can select the analyte when their chemical structures differ from each other. So, selectivity of mass spec is pretty good to distinguish the focused analyte from the other's impurities. The last reason is structural elucidating power. Structural elucidation capability is one of the most important features of mass spec. In mass spectrometer, we can make unknown analyte collide with inert gas molecules. By the collisions, the unknown analyte will break down into small fragments. Because fragmentation reactions must occur according to the structure of intact unknown molecule, the reactions imply the chemical structure of unknown. For example, in the case of mass of unknown was measured to determine its chemical formula as C5H6N2O, and then broke down the unknown step by step to obtain small fragments, C5H4NO and C5H4N. This unknown may have one pyridine ring, OH, and NH2. This is the very basic concept of structural elucidation on unknown by using mass spec. The detailed way will be explained in another video. This is a table of major methods for structural elucidation of trace unknowns. Infrared spectroscopy, IR, nuclear magnetic resonance spectroscopy, NMR, and mass spectrometry are the major methods. Their analytical principles are molecular vibration, nuclear magnetic moment, and mass-to-charge ratio. Usually, we may reveal unknown structures by using IR and NMR. These spectroscopies are basic and powerful, to obtain information about which kinds of functional groups does unknown have in it, and the detailed information of its chemical structure. But they require individual pure compounds, as they are not available online separation techniques. On the other hand, mass spec does not require individual pure compounds, and is acceptable to analyze complicated mixture. This unique feature is highly important, because the isolation of individual pure compounds from environmental samples is almost impossible. That is why mass spec is the most suitable elucidation method for environmental unknowns. This is the summary of this video: why mass spec is the most suitable elucidation method for environmental unknowns. There are 3 reasons: sensitivity to detect trace contaminants in the environment such as 10 femtograms in 1 microliter. Selectivity to distinguish resemble contaminants based on different masses or different structure. And structural elucidating power of unknown contaminants in the complicated mixture. These 3 features are indispensable for environmental chemistry. Also, these features may be useful in your own research besides environmental chemistry, like biology, biochemistry, pharmacy, organic or inorganic chemistry, food safety, and so on. Thank you for watching.